It's my pleasure to introduce Volker Thiel. Volker. Volker, if you could come on up. I've known Volker, or we've known each other since I think we were PhD students working on coronaviruses, me on pig coronaviruses. Volker, what were you on?
Mouse hepatitis
virus. Mouse hepatitis virus, right? Those are the obscure coronaviruses we were working with. And Volker has stayed with coronaviruses. I was a Johnny come lately. I left them and moved to negative strands, which is why it's been great to invite Paul Dupre to give another keynote, because I know Paul swapped strands as well. But Volker has always been a coronavirologist, and has done some really instrumental, innovative work on the host cell interactions, but particularly the molecular virology of viruses. So Volker, thank you very much for accepting our invitation to come and talk. You have to just put that around the back. And we look forward to hearing what you have to say. So yes. Thank you, Volker.
Thanks, Julian. So thanks a lot. It's great to be here. And as Julian said, there is a past history before, the pandemic of coronaviruses that met in early days working on strange viruses. And it was very nice to see during the pandemic that all those connections that the young researchers may have established during the pandemic, that all those connections that we had before could be revived. It was very great to see that this network was emerging, and we also received some help for the International Nidovirus Symposium from the network. So many thanks for that. And it was really nice also to be, there was Julian, you didn't mention the Cyprus meeting. So that was also a great opportunity to be on site on a outbreak of an animal coronavirus. So today I want to talk about some basic research we are doing, or what we have been doing for many years, and also how to translate it into something that may help us in situations like this pandemic. But before I go to, okay, here we are. Of course, for being the first time in Liverpool, I was driving in with a taxi driver, and he told me this is the place to go. And I went in there. I met Lisa here. So Lisa is over there, and her friend Vicky that studied here in Liverpool actually many years ago. But I heard from the taxi driver not only that you have to go in, you have to really go for the real sightseeing. So that's the thing. That's the thing you need to. I did all that, and then in the afternoon we went to another place because we are big football fans, and of course had to go to Anfield and make a tour there. And the highlight, of course, was then last night, England winning against Italy. So we're very into that because it's currently taking place in Switzerland. So there's a huge hype in Switzerland going on about football. All right, so go to some more serious. So SARS, Julian already mentioned there was something like SARS-1 in 2002, 2003, which also caught the attention of the media, not as much as today. And also we didn't see so many cases. Retrospectively, at the time it was a big thing, 8,000 cases. 800 deaths almost, and interestingly, the virus made it to 30 countries. So already at this time, how we travel, how we move, how we behave had an impact on the spread of this virus globally. But as you can see from this graph, these are the case numbers in China from the end of 2002 to mid 2003. So it was. They could contain the virus, and that was mainly because the virus was not spreading or transmitting before, or in many cases before the disease was visible. So you could spot the symptoms and then isolate people and then contain essentially the virus. So that was the big difference compared to SARS-CoV-2. But it was, of course, interesting to see where did the virus come from. So there were many studies going out, sampling in the wild. Up to then, this is a table from 2004. We knew some coronaviruses, animal coronavirus, but all animals, that have to do something with us, livestock, companion animals, not really any viruses from wild animals out there. So that was our perspective at the time. Human coronaviruses, we knew seasonal ones, 229E, OC43, NS63, interestingly, was discovered only in 2004 after SARS-1, and people really looked for coronaviruses. Are there other coronaviruses in humans? And they found them immediately, more or less. And they were, they're globally distributed. The typical seasonal coronaviruses, common cold viruses. So NS63 was discovered in 2004, HK1 in 2005, and they all have always been there and were just not detected because we didn't look for it. But it then became apparent that these are, the coronaviruses we knew before, essentially in this phylogenetic tree, and all those are from bats. So these are sequences found in bats, and that really gave rise to the idea that, first of all, zoonotic transmission must have happened before. We have close relatives in bats. That's a paper that is actually from Christian's group that showed that 229E has close relatives in bats. So obviously this virus is originating from a virus that was probably in bats before. And it came most likely through camels and then to humans. So this was obviously not a new thing with SARS-1. There's a huge diversity in bats. You see alpha coronaviruses, beta coronaviruses. So there are many different viruses in those animals. And when you look at SARS-related coronaviruses, you see here the Wuhan SARS coronavirus 2 virus and the Frankfurt 1 SARS coronavirus 1. So this phylogenetic gap can easily be filled by viruses that have been detected in bats, mostly bats. So also in pangolins, but mostly in bats. So these viruses are out there and they recombine. So if you look at the genetic makeup, this is SARS-CoV-2, and if you look at those viruses that are related down to SARS-CoV-1 and just color according to sequence identity, you see that some are very closely related or identical almost until some regions that are less identical. And this is a typical mosaic-like structure or pattern that indicates that there is recombination. So these viruses are not only out there, they also recombine and may gather. You see here that the spike protein is obviously a hotspot for recombination, maybe not for the mechanism of recombination, but for the selection. So with the spike, you can gain easily access to new hosts, get a new tropism, and that's maybe what is driving that. So this was actually known before. When we look at this timeline again, I mentioned seasonal coronaviruses. Julian mentioned, of course, MERS and then SARS-2. Infectious bronchitis virus is an animal. Yeah, you know it's Julian. It's discovered in 1931. So coronaviruses are known for long. And if you look at what has been done in terms of the molecular biology, we have seen the first sequenced genome, the idea or the concept of polyprotein processing, how that works in coronaviruses. Then the first attempts to generate recombinant viruses by targeted recombination. And then finally, the full-length reverse genetic clones. So that was in 2000. and marked essentially, the start of the reverse genetic era, where we could really knock out specific functions in the genome and see what those functions, what they impact on. And that gave rise to several studies where we also were involved to characterize enzymatic activities of the replicase complex primarily, but also all the accessory proteins that we didn't have an idea what they are exactly doing. You could see that they are impacting on pathogenicity, and this knowledge all helped us to get, when SARS-CoV-2 came, to already have a little bit of an understanding how this virus works. So this was the time 2000 when we were assembling the clone. This was the way how we communicated, how we displayed our research. But we were always interested in improving that because it was still a little bit cumbersome, time consuming. And this was a project we had before the pandemic, HONORS, a European project with a consortium of several labs headed by Leah van der Hoek in Amsterdam on pandemic preparedness. And our part of this consortium was to establish a reverse genetic system that is fast and versatile and can be applied to many viruses. And we tried that because Jörg Jores at the time, Institute of Veterinary Bacteriology, he came to Bern and brought a cloning system with him, a cloning system in yeast. And this was so interesting for us that we thought, okay, we try if that makes sense for our purposes. And with Tao and Nadine, and together with Fabian from Jörg's group, we set up the system. So it's very simple. You have DNA fragments that are overlapping, covering a viral genome. In this case, we tried it first with mouse hepatitis virus, already having the GFP inside to easily see it. And we did just PCRs that overlapped, and you have to generate an overlap at the termini to the cloning vector, the plasmid backbone, and then you just put those fragments into yeast. And yeast is doing the magic thing of recombination and makes this circular DNA. And then you can go from there to prepare your DNA, your in vitro transcript, which is generating essentially or representing the viral genome, put it into cells and collect the supernatant. And this was actually quite efficient. So the first clones we randomly picked were working, and we since then worked with this system. And this was in 2019 when we had essentially extended to see whether other coronaviruses work as easy as the mouse hepatitis virus, MERS coronavirus, 229, human coronavirus, 229E, HK1, and so on. Also beyond coronavirus, Zika virus, and human RSV-B. And this was just from a swab. So it was also possible just to PCR out the fragments from a clinical sample and directly go into making a clone. And then this was at the end of 2019. We were ready to write our paper up, and then something happened, as you know, in 2020 or at the end of 2019 with the pandemic. In January 10th, on January 10th and 11th, the first sequences were reported, full-length sequence of SARS-CoV-2, and that of course was the starting point for us to clone the virus in order to, because at that time nobody had the virus in the lab. It was still everything more or less in China. There were outbreaks outside China starting, but everybody was waiting, let's say, to get the virus to start to implement diagnostics and all these things. And we ordered immediately synthetic DNA fragments covering the entire virus. And then after receiving them, this took about 3 weeks to have all fragments. We still had a gap because one fragment was not easy to synthesize. But luckily, also Christian helped us out in sending us the first supernatant of the very first culture they made from the Munich case at that time, so that we had some material to PCR the fragment and then fill this gap, and then it was very straightforward within 1 week that we could clone the virus, and that was then in early February. And I just want to point out that in Australia, end of January 2020, this was the first time when a virus could be isolated outside China. So this was the first time when, and they of course were very busy in growing the virus and shipping the virus to other labs. So with this system, of course, we have now also started to do molecular work. And there was also something else before the pandemic where we went already into a direction of looking at translation. So I sometimes go to schools and to explain, the kids how viruses work, what they are, how they look like. And one thing I always point out is that, in our cells we have essentially a map or a plan. We have, the building is, let's say, the proteins, but we have to have the workers that read the plan and construct the building. Viruses are the same. They have the plan, they have the building, but they lack essentially the man that can construct their virus, so they are dependent on translation. So that's the very central point of all viruses, that they are dependent on translation. And apparently they have found many ways to interfere with translation. And we came to that a little bit by a screen, and we didn't aim to go into this direction. But a screen is like a box of chocolates. You never know what you're gonna get. And so we were essentially looking at the time or at the replicase complex to look at the proteins that are in close proximity. So we did that by adding biotin ligase as a fusion protein to the replicase complex. And then we could essentially, by proximity labeling, detect all the proteins that are in close proximity to the site of RNA synthesis in the cells. So this is how it looks like. So this is the stain where we have placed, the NSP2, where the NSP2 is where we have placed the biotin ligase. So it is at the sites of these double membrane vesicles where RNA synthesis takes place. And what we found is, of course, all the non-structural proteins, part of the replicase complex as expected. But one cluster translation was very, very dominant and really stood out as a very interesting cluster. So many factors for translation that were also highly significant in this screen. So this was also published in 2019. And as you can imagine, we didn't have time then afterwards to really mine this data set. But we are going back to that. Another piece of data, this is a work from Reto Lang in our lab. So he is looking at stress granules. So stress granules are, granules that arise when there is stress in the cell, and they are essentially composed of stall translation complexes. So ribosomes are mRNA, with the idea to calm down the cell a little bit, to stop or reduce translation so that the cell can recover from the stress. And we see that here with mouse hepatitis and Semliki forest virus, that Semliki forest virus makes, this is already known, very early on stress granules. We see them with mouse hepatitis virus much later. But it's not that the viruses have different kinetics, because they have their peak titers around the same time, more or less. It's just that Semliki forest virus has the stress granules much earlier. They resolve it, and one protein is obviously involved in actively resolving them, and they obviously need that at the beginning of the exponential growth phase. While with mouse hepatitis virus, it comes much later. And we also found, which was quite interesting, when we labeled for viral RNA, which should also be in the stress granules because it's also mRNA. You see here, if you look at this black hole here, that's a stress granule. So it has no viral RNA in the stress granules. So that was also very, very intriguing. You can also go more into an analysis here that you see the stress granules, but no viral RNA. So obviously the virus is taking care very much about translation. That the mRNAs the virus is producing are favored over the cellular mRNAs. What we also started now to look at is the very early phase of infection. Essentially, when the virus is entering, it's delivering one copy of the viral genome, and this is getting translated. And there is now a system available that can visualize this process. So it functions very, very simple. You have an array of epitope tags. So we introduced that again into NSP2 in the mouse hepatitis virus genome. And then once this genome is being translated, the epitope at some point is being translated, and each epitope can bind one molecule of a single chain antibody fused to GFP that is expressed in the infected cell. So once you have this cell line expressing this antibody, you can nicely monitor the translation of this single RNA molecule. So we have 6 copies of this epitope. So 6 copies of GFP plus it's not only one ribosome that is translating the polysomes. So there's quite a number of GFP molecules on one spot, which you can easily see then in the cell. And this looks like this if you have an infected cell line. So the virus is taking over very quickly within the first 1 to 3 hours, and then it's already more or less saturated. Also, if we go a little bit closer. And essentially what we see is within minutes, we see the first viral genomes in the cell being translated. So you can actually really see the incoming genome that is being translated. Then after about an hour, you see more foci of fluorescence. So obviously one round of replication has occurred, and the infection was successful, and then the system is already saturated. You can also use that for, I mentioned, recombination with SARS-CoV-2. You can also use that system for studying co-infection. So here we have infected with 2 viruses, having 2 different epitope tags, and we have 2 different labeling systems to get red and green signals. And then you see the red and green infecting genomes, how they multiply, and at which point they come into proximity, which is of course important for recombination. And just to compare that with a standard, insertion of a GFP in the replicase complex, so the sensitivity is just much higher. So we see it in the first minutes, and here we need to wait a couple of hours until enough GFP has accumulated. So this system gave us a nice idea what happens in the very early phase. We are exploring this at the moment. We can see the incoming genome. Interestingly, many infections appear to be abortive. So you see a translated genome, but then nothing else happens. So there is obviously a way, either the genome is defective or the cell has a word to say at this stage to maybe stop the infection. Also, accordingly, the number of translation events we see from directly after infection exceeds the number of infectious viruses. So when we titer the virus And we all know that infectious titer is much less than the genome or number of genomes. So there is a difference, and it looks like many of those viruses that are maybe that we can detect by PCR as genome equivalents can make it actually into the cell, but stop for some reasons. We can also look at co-localization of non-structural proteins, but ultimately we would also go into looking at co-localization of replication and transcription complexes. So if two viruses get together in one cell, are the replicase complexes clonal? So each complex is from one virus but doesn't mix with parts from the other virus. So these basic questions. We think overall that this is the first hour or two is a very vulnerable phase for virus infection, because it's maybe one genome and the cell may have still a possibility to interfere and stop the infection at this point. So there are maybe many host factors or restriction factors that come into play here. All right, so translation, as you see, is a big thing in our lab since a while. And we also, at the beginning of the pandemic, thought, okay, we also would like to use what we have learned in order to, in this case, generate attenuated viruses. So what we have established in the beginning of the pandemic is a network of people and labs that enable us really to go from the start of making a recombinant virus to complex in vitro systems and in vivo systems to study SARS-CoV-2, but also to develop vaccines. And we work together here a lot with Martin Beer at the FLI in Reims in Germany, Klaus Osterrieder in Berlin, Ramesh Pillai in Geneva. So he's a expert on RNA modification, and Sebastian Leitner, expert on translation at the University of Bern. And we had a couple of ideas in the beginning, just on paper, how attenuated viruses could be generated. And there are two concepts that are already known. So one is known for many years, codon pair de-optimization, introduced by Eckhard Wimmer many years ago, where you essentially have codon pairs that appear to have a very low frequency. For some codon pairs, the expected frequency would be just equally distributed is much lower than expected, and that may have steric reasons, during translation, that the codon pairs, that the amino acids may not be easily translated in a certain context, or and the idea here is really to enrich the viral or the codons for those who are rare, or codon pairs that are rare, and this obviously attenuates the virus. So the exact mechanism why this is working is still not known, but it's very well established that this is working. The other one is a one-to-stop codon strategy, was introduced by Marco Vinuzzi's lab. So you enrich the virus by codons that only need one more mutations to get to a stop codon. So there are many synonymous codons that for certain amino acids that need two mutations to get to a stop codon, and you change all those codons to synonymous codons that only need one more mutation. And then by the error rate of the polymerase during replication, you generate genomes with a higher likelihood that have a stop codon in this region. And finally, modifying translation kinetics. That's something that was not yet described or has not yet been described. So the idea here is to use ribosomal profiling, where you essentially look in infected cells at a certain time and prepare the ribosome fraction that is bound to the RNA. Then you digest the RNA, so everything that is not protected by a ribosome will be digested, degraded, and then you end up with short reads that represent the sequences that were just within the ribosome at the time when you did the analysis. So those codons essentially that were translated at this time point when you did the preparation. And this gives you an idea about what is being translated in a cell if you look for cellular mRNAs, but also for viral RNAs. And then you can see also whether certain codons are translated with a high or a low speed. So those who are represented more, let's say, so it's more likely that you catch a ribosome on them are probably the slow ones, because the ribosome may sit longer on those codons and then they are more represented in the library. And those who are less represented are the ones where the ribosomes are going fast in translation. And then you can do the same and look for synonymous codons. In this case, we made a ranking from slow to fast and made then recoded viruses in order to make slow or fast viruses or fast genes, let's say. And we did that in a couple of regions. So one was fast, so spike, membrane, envelope, and nucleocapsids, all the structural proteins. And we also made a slow one, but couldn't recover that or rescue that. So that's obviously too attenuated. But the fast one we could get. We also did a slow and a fast spike version. And then we also did something, you know, within the open reading frame 1A, 1B, there is this ribosomal frameshift site. So a huge RNA structure where ribosomes have to overcome, that ribosomes have to overcome. So there might be that's sort of a barrier for them. And what we did is to make a fast region just before the frameshift site, so making a highway, and a slow region after that, so making a traffic jam. So the ribosomes come in full speed, and then they see all the slow codons and queue up at this point. So that was the naive idea, but obviously it worked well because those viruses are really crippled, so they make only tiny plaques and seem to be very attenuated. So these are, you know, short summaries of viruses, growth curves of those viruses. They all are to some degree attenuated. The most is this fast slow version around the frameshift site. And we also went to look for, the abundance, for instance, of the spike protein. We think we have to still calculate that in detail, but we think that the slow spike also generates viruses with less spike on the surface. So obviously there's less protein, as indicated here in the plot. And we also looked at their attenuation in the K18 humanase 2 mice, and they are all attenuated. So that's sort of proof of principle that this system is really also useful for attenuating viruses. The other, and maybe I use that for the last minutes. The other system was the one-to-stop strategy. You can see here again how that was done to change a codon that needs two mutations to become a stop codon to a synonymous, where only one mutation then gives rise to a stop codon or can become a stop codon. And we recoded several regions. So the fragments essentially, as we use them for cloning, so fragments 4, 5, 7, and 8. So in the 1A and in the 1B regions we have chosen. Because in this region, when there is a stop codon, obviously the virus is dead because they need the replicase proteins. And we have overall modified around 300 codons, about 600 nucleotide changes, and the overall region of those fragments is about 11 kilobases. Interesting with this, you can essentially adapt the level of attenuation. So depending on how many codons you change, so how big the region is you are recoding. So for instance, when we have made viruses where only 2 fragments were recoded, or 5 or 7, 8, in this lethal mouse model, already 50% of mice survived. If we combine all 4 fragments, then all the mice survive when they are infected. So you can really see how, increasing the number of codon changes, how this adds up to increased attenuation. And also here, you see the body weight, that those viruses didn't change much in terms of also the body weight. You can also look in the animals. So in this model, of course, you can look at the lung and stain for virus, but also it's in this model the virus is going rapidly to the brain because of overexpression of the human ACE2, and you can nicely see that with a 2 fragments modification, there's already a great reduction, but still there is virus. With the 4 fragments, you barely see anything. So it's a very clean system. And of course, since we are coronavirologists, we also want to add something on top of that. So we also deleted in a vaccine candidate now the accessory genes of 6 to 8, and we have modified NSP1. This has been described to essentially bind to ribosomes at the mRNA entry channel to inhibit cellular or translation of cellular mRNAs, and we have modified the residues, amino acid residues that are needed for binding to the ribosome, so that this mechanism of blocking mRNA translation is also released. And with this, we came quite far. So here are a number of candidates that are all, protecting. So this is a challenge experiment. So they don't lose any body weight after challenge. Also, they all survive. And we also compared that with mRNA vaccines. The one example I'm showing here. So we did immunization intranasally with the live attenuated virus, and intramuscularly with mRNA, as it was done with most of us, I guess. And then after 28 days, we came with a challenge, in this case Delta, which is the most virulent variant we have seen in the pandemic, and then looked at 2 and 5 days in the animals, and we particularly looked at the lungs. And here you see the stain for the nucleocapsid. You see that 2 days post-challenge, there is virus in the lungs. There's more virus, obviously, in lungs of mRNA immunized animals. But at 5 days, it's mostly resolved. You still see a little bit of virus. What you can do here now is to maybe I go back to do spatial transcriptomics. So you can look at the sites where the virus is and then see what is expressed at those sites. So you see cellular but also viral transcripts. And then you can see here, for instance, if you look at the orange ones, this is 2 days post-challenge for the live attenuated vaccinated animals, and 5 days post-challenge. You see the viral transcripts. You can easily detect them for at 2 days, but almost not anymore at 5 days. While with mRNA, that are the purple ones, at 2 days you can detect much more, as we have already guessed from the images, and you still can detect transcripts after 5 days post-challenge. But the interesting thing is, of course, to look at the cellular response. And here, not surprisingly, the JAK-STAT pathway stood out as the response, the host response we see at the site of viral replication in the lungs. But also here you see that There is upregulation of these pathways 2 days post-challenge, but it's almost back to baseline, which is here after 5 days post-challenge in the mice that received the live attenuated vaccine. While with the mRNA vaccination, you can still see upregulation of this pathway after 5 days, so it takes longer for the mice to resolve the infection. And the last piece of data we have on that I can share today is we also deleted the famous furin cleavage site in the spike with the idea that and Andrew has shown that very early in the pandemic that in cell culture you lose this very easily. And we aim to produce this vaccine, of course, in a regular cell line, and we were expecting that this vaccine candidate will lose the spike cleavage site anyway during production, and this should not be, because you should have a stable vaccine. So we deleted it with the idea to have it stable through the processing or production phase, and then what we saw, which was sort of a bonus, if you just look at the yellow dots here, this come from this candidate that has, in addition, the deleted cleavage site. That the deeper you go into the lung, you see less replication. So it's obviously even safer with this deletion. And most importantly, we also vaccinated animals, and then this was all done in hamsters, and then after they put naive animals into the same cage to see whether the vaccine is transmitted, is being transmitted to the contact. And you see that it's transmitted for all our candidates except the one where the cleavage site is missing. So here the contact didn't seroconvert. There was no indication that this vaccine was shed in this setting. With this, I'm at the end. I hope I could convince you that translation is important, is an important mechanism, particularly for viruses. They're all dependent on it, and that we can use some of our knowledge to make use of that. I mentioned it already in the beginning, so many thanks to the network for supporting us at the nidovirus meeting symposium in Montreux. And I also want to announce, or it's maybe not me who should announce, Leo Poon essentially, but he already gave me a brief message on the date, so 24th to 27th of June next year in Hong Kong. And I hope he will make a proper announcement very soon about the next nidovirus meeting. And with this, I stop here. I think many people from our lab. Kim will speak in the next session about our work, or part of our work, on feline coronaviruses. And I mean, I think many people in different projects. So Martin was doing the imaging, Reto was doing the stress granules, Nadine, Tuba were doing most of the in vivo work with the vaccine. Bettina was doing essentially all the cloning, Ettore with her fast and slow codons and so on. And Lisa for helping me keeping all things in order. So, and all the collaborators, I already introduced them during the talk. And with this, I'm stopping here. Thank you.
Thank you, Volker, for your interesting talk. Do we have time for a quick question? Yeah, any questions for Volker?
Yeah, it's live. With the one to stop strategy, can you actually see evidence of increased stop codons in minor variants when you sequence the virus?
We didn't do that yet. What we see is that they are more vulnerable to mutagenic drugs. So like molnupiravir. Yeah. So they obviously accumulate then more mutations, and this is, for those viruses, more has a more impact than for the wild type virus. But we haven't gone into deep sequencing, you know, the virus stocks.
So, David, sorry, if I could just follow on from Paul's vein, Volker, lovely work. The sort of following on from Paul's question, when we deep sequence normal viruses like Ebola or SARS-2 or whatever, you do see full length. I don't know whether they're full length, but you see defective genomes with a number of stops in. Do you think if you look at the viral genome and actually then do the opposite experiment and engineer out the things that could easily change to a stop, you would not improve, that's the wrong word, but the pathogenicity of the virus would increase? In other words, do you think there's a natural mechanism in the viruses to regulate, to attenuate themselves through this mechanism that you mentioned? Thank you, Bobby, for that question.
Yes, that goes in towards, you know, a question that is always coming, what is the real mechanism for the attenuation? Because a few viruses that drop out because they have a stop codon may not do the job. There are several ideas, but nothing is really shown in mechanistically. So I think what happens is, or what could happen is that many of the RNA repair, RNA degradation mechanisms are turned on by those defective viruses. You have seen that also, you know, when we looked or when we dived into the replication translation speed, obviously there is also some adaptation for the virus. So the virus is adapting, you know. For instance, we had the feeling, but we have to look into that before transmembrane regions, there is tendency to have slower translation to allow maybe folding of the nascent protein chains. So there are many things you may disturb with this strategy that are maybe not, you know, because there is a stop codon. But overall, I think it may work in a way that maybe several of those repair or RNA degradation mechanisms are turned on, and then followed by sensing of viral infections and making the cells aware earlier than maybe normal that a virus is replicating.
Thanks, Bob.
Thanks. Dave, if we just head to you and then we'll move on to the next speaker. Yeah.
Volker, that was really cool. Can I ask, the mechanism of attenuation, again, of these, maybe not the one to stop, but where you de-optimize the codon pairs or the codon usages, your data suggested that there is sort of a slowing down and a down-regulation of translation, the Western blots with Spike, for example. But other papers suggest that this is more sort of CpG content and innate immune activation.
Exactly.
Is this the same pathway being sensed differently, or are they two different pathways that are both contributing?
I can only speculate, because all of what you said has been discussed and is sort of shown that these are possible mechanisms. But one really would have to design or recode the genes in a way that they exclude the different mechanism, which is very, very difficult. And we didn't have the time in the beginning of the pandemic to really look at that, because we just assumed it will work, and whether it works by this or that mechanism is maybe not so important. But that would be a very interesting question, because at the end of the day, we would like to really know why things are working, and also we also see, you know, for different recoding strategies a different impact. So we need, for the one to stop, approximately double the numbers of codon changes than we need for the slow fast or the codon pair de-optimization. So there is a clear difference, but we cannot tell at the moment what the mechanisms are.
Okay, thanks.
Yeah. Thank you very much, Volker. That was great. Thank you.